Welcome to the Explainer. I'm absolutely thrilled you're here today because we're going to explore what might just be the biggest plot twist in modern biology. Now, you know the drill with CRISPR. For years, we've celebrated it as the ultimate pair of molecular scissors, right? A hyper-precise tool to edit genomes, cure genetic diseases, the whole shebang. But today, we're taking CRISPR beyond the lab bench and way out of the editing room. We're going to see exactly how scientists flipped the script and transformed this famous gene editing tool into a hypersensitive disease detective. But to really understand this transformation, we have to start with kind of a wild question. What if CRISPR's biggest flaw was actually its greatest feature? Think about it. We usually hype CRISPR up for its surgical precision, its ability to find one exact sequence in a sea of literally billions of base pairs and cut only there. But get this, its incredible power as a diagnostic tool actually comes from a spectacularly messy, non-specific behavior. I know, it sounds totally counterintuitive, but stick with me. By the end of this explainer, you'll see exactly why this perceived flaw is completely revolutionizing medicine. So here's our game plan for today. We'll uncover CRISPR's unexpected plot twist, dive into the mechanics of collateral cleavage, and then meet Sherlock and Detector. After that, we'll walk through the diagnostic workflow, look beyond infections into oncology, and finally weigh the real-world advantages and limitations. All right, let's get right into Section 1, CRISPR's unexpected plot twist. So the foundation of this entire field really rests on a phenomenon known as collateral cleavage. Sometimes you'll hear it called transcleavage. Here's how it works. When the guide RNA successfully binds to its exact target sequence, it triggers this massive conformational change in the Cas protein. It's kind of like imagine a highly trained guard dog that finally catches the specific intruder it was looking for, but then suddenly goes completely wild and starts shredding everything else in the room. That's literally what happens. Once certain Cas proteins bind to their specific target, they just hyperactivate. They turn into these non-specific molecular shredders, indiscriminately destroying any nearby single-stranded nucleic acids. Now, for gene editing, that would be an absolute nightmare, right? But for diagnostics, it turns out to be a highly controlled, absolutely brilliant signal amplification engine.
Which brings us to Section 2, the secret, collateral cleavage.
Okay, take a look at this elegant three-step process we've got going on here. First up, the Cas protein and its guide RNA find their specific target. Let's say it's a piece of RNA from a virus. Second, it performs a cis cleavage, which just means it cuts that specific target. But here is the crazy part. That act of cutting throws a molecular switch, and that triggers step 3. The transcleavage shredder. So to capture this chaos, scientists add millions of reporter molecules to the mix. These reporters have a bright fluorescent tag on one end, but it's kept dark by a quencher on the other end. When the Cas protein goes into its hyperactive shredding mode, it slices these reporters right in half. The quencher gets separated from the fluorophore, and bam, suddenly the whole sample glows with a bright, visible signal. A tiny, tiny input generates this massive, glowing output. It's genius. Now, let's move on to Section 3, meet Sherlock and Detect. Here we go, the heavyweights. On the left, we've got Sherlock, developed by the Zhang Lab at the Broad Institute and MIT. And on the right, Detector, developed by the Doudna Lab at UC Berkeley. Honestly, it's a fascinating continuation of that historic CRISPR rivalry between these two pioneering labs. Now, while both of these platforms rely on that collateral cleavage phenomenon we just talked about, they actually have entirely distinct molecular personalities. They use completely different Class II CRISPR effectors to get the job done. And notice the difference in their approach to amplification, too. Sherlock relies on a pre-amplification step using RPA, while Detector utilizes LAMP. So they're basically distinct, specialized tools tailored for very specific jobs. So if you're going to remember one crucial thing today, let it be this golden rule of thumb for diagnostics. Sherlock uses a protein called Cas13, and it is specifically designed for RNA detection. That means its collateral shredder destroys single-stranded RNA reporters. Detector, on the other hand, utilizes Cas12a. That targets DNA, and its collateral activity destroys single-stranded DNA reporters. It's pretty simple when you break it down. If you want to find an RNA virus, you call Sherlock. If you're hunting for a DNA target, you call Detector. All right, let's see how this actually plays out in a clinic in Section 4, the diagnostic workflow. The true beauty of this technology is how incredibly streamlined the clinical workflow is. Step 1 is super straightforward. Just extract the nucleic acids from a patient sample, like a nasal swab or a blood draw, using a basic lysis buffer to break open the cells. Then we hit step 2, and this is where the magic really starts: isothermal preamplification. Now, unlike traditional PCR that requires those bulky, expensive thermocyclers to heat and cool the sample over and over again, techniques like RPA or LAMP just copy the target DNA or RNA thousands of times at a single constant temperature. It's brilliant. Finally, in step 3, our Cas13 or Cas12a proteins are introduced. If that amplified target is present, collateral cleavage is triggered, the reporters get shredded, and you get a crystal clear result. You can read it on a fluorescent reader, or even just a simple paper strip that literally looks exactly like a pregnancy test.
Moving right along to Section 5, Beyond Infections, Oncology.
Okay, take a second to just really look at this staggering number, 10 to the negative 18th molar. Wow. This is what we call attomolar sensitivity. But what does that actually mean for you and me in the real world? Well, it means that Sherlock and Detector can detect literally a single target molecule in a microliter of a sample, just one. To put that into perspective, it's the molecular equivalent of finding a specific grain of sand in an Olympic-sized swimming pool. I mean, it's just mind-blowing, and it's the absolute key to why CRISPR diagnostics are such a game changer. And with that crazy attomolar sensitivity, the real-world impacts are just dramatic. Let's look at oncology. We know tumors shed these tiny amounts of DNA into the bloodstream, right? Well, CRISPR diagnostics can detect this circulating tumor DNA at concentrations as low as 0.1 attomolar. That means we could potentially catch cancer extremely early, just through a simple liquid biopsy blood draw. And in infectious disease, it can pinpoint antimicrobial resistance genes in under an hour. That allows doctors to deploy the exact right targeted antibiotics to stop sepsis fast. And honestly, this next part is incredible. It's so precise it can even distinguish between pathogen strains that differ by just a single nucleotide. Think about that level of resolution for a second. It's like spotting a one-letter typo in a book with millions of words.
Let's wrap things up with Section 6, Advantages and Limitations.
Really, CRISPR diagnostics act as kind of the perfect Goldilocks solution. If you compare these columns directly, you'll see what I mean. RT-PCR is obviously the gold standard for accuracy, but it requires expensive thermocyclers and takes hours in a highly trained lab. Rapid antigen tests? Sure, they're fast and cheap, but they fundamentally lack that deep sensitivity. CRISPR, though, gives us the incredible attomolar sensitivity of PCR, but with the minimal equipment and speed approaching a rapid antigen test. And because it operates at a single temperature, isothermally remember, it holds massive potential for point-of-care testing in remote areas that entirely lack laboratory infrastructure. But you knew there had to be a catch, right? We need to be analytical here. The technology isn't completely flawless yet. Achieving that mind-blowing attomolar sensitivity absolutely relies on step 2 of our workflow, that preamplification step using RPA or LAMP. Because without preamplifying the sample first, bare CRISPR detection is really only sensitive down to the picomolar range. And frankly, that just isn't sensitive enough for most clinical diagnostics. Plus, unfortunately, adding this preamplification step introduces a major risk of carryover contamination. That's where amplified products from one test could accidentally contaminate the next, causing false positives. It's a significant clinical hurdle for sure, but researchers are actively working to overcome it as we speak. Which brings us to our final thought for today. We started this whole journey with a tool used to edit genes in a lab, and we've ended up with a molecular detective capable of attomolar precision, all powered by the brilliantly chaotic mechanism of collateral cleavage. The transition from genetic scissors to diagnostic shredders is just nothing short of incredible. So I'm going to leave you with this. If CRISPR can already find a single cancer mutation or a hidden superbug gene in a single drop of blood or saliva, what on earth is it going to uncover next? The diagnostic revolution is honestly just getting started. Thank you so much for joining me on this explainer, and keep questioning what's possible.